Alright, so we are going to be going through a series of kind of online lectures where, one second, let me just make sure that this thing, okay, good. Sorry, I thought my PowerPoint was gonna automatically progress to the next slide. We are going to be going through a series of lectures on a number of economically, particularly agriculturally, important plant families. Unfortunately, we cannot do this series live in person because of the pandemic, so we're doing this remotely, and so I am starting to aid in the process of actually getting things recorded and set up. So I've made a large PowerPoint that goes through about 50 of the major plant families that humans use extensively, mostly for food. Now, it's not to say that farming and the production of plants is only about producing things to eat. However, we are going to stay with families that have at least one species that we use for food. So the only ornamental families are not going to show up here. However, there are plenty of families that have members that are used both ornamentally and agriculturally. And the families that are useful for non-human food use but agricultural importance, such as plants necessary to feed livestock or to produce different chemicals to use on farms or for nutrient return cycling, for example, we are going to run through. So before we get totally into the families, I'm going to briefly touch on why we care about the family level and kind of where the whole family level comes from. And so there's a field called taxonomy, and this is the study of how organisms relate to one another. Nothing exists in a vacuum, and so organisms are going to have varying degrees of relatedness or non-relatedness to each other. Taxonomy is concerned with giving everything names, putting things in categories. And so some of the ways of looking at the groups that these organisms belong to are based on taxonomy. For people wondering how we have an idea of the placement of things, the evolutionary processes behind organismal diversity is a good place to start, and we're cued into these things mostly by morphological features. Embryological study is also important. This is more pertinent to the animal world than the plant world, and these are by no means the only two groups of organisms we concern ourselves with. However, in the animal world, the early life stages of most vertebrates look incredibly similar, and the closer two organisms are in relation to each other, the longer their embryos, fetuses, and even young offspring look nearly identical. So most birds, for example, are going to have a general body shape and body plan in the egg that there's really no deviation from until a certain point. And birds that, perhaps have very long necks will diverge from this plan sooner than they would diverge from one another. So ostriches and flamingos would look more like each other for a greater proportion of their development in the egg than an ostrich would look compared to a hummingbird embryo. Anyways, now we test the DNA of organisms because we have that technology. One of the things that's important to know in evolution is that things are conserved. DNA sequences are not totally rewritten, so the closer two organisms are to one another in relation, the more shared DNA they should have. And we look at a number of different parts of the DNA that sequences different cytochromes. Cytochrome C is a very popular part of the DNA that is used to kind of determine how close two organisms are to one another. And because we have convergent evolution, wherein similar appearing structures or similar functions can be the result of a similar environment, even though the organisms that feature them are not closely related whatsoever, we now rely heavily on DNA evidence because we have made a lot of taxonomic revisions that we wouldn't have been able to make if we were just going with morphological evidence, which is what we had back then, but now we have a better way of looking at these things. So we are going to concern ourselves with the levels of taxonomy at which some of these groups fit. So it goes from domain, which is going to either be prokarya, eukarya, or archaea. Now, we're not going to worry about the prokarya or the archaea because all of our organisms are going to be plants, which are eukaryotes. This has to do with the cell type. Eukaryotic cells differ from prokaryotic cells in that their DNA is bound by a membranous nucleus, whereas prokaryotes lack this. There are a few other differences between prokaryotes and eukaryotes, but we're not going to get into that because it's beyond the scope of what we're doing here. Then you have kingdom. So we're going to stay within the plant kingdom. Phylum, which is a level down, and phylum is mostly to discern, I don't know how to put it. These are groups where you hone in, and there is a rule in evolution where you don't outgrow your ancestry. So once you're a eukaryote, you stay a eukaryote. Once you're a plant, you stay a plant. Once you're in a particular plant phylum, which we are going to stay within the angiosperm phylum. Angiosperms are amazing. You stay an angiosperm, and so on. Then we get to class. For plants in the angiosperm phylum, we are concerned mostly with monocots and dicots. Now, remember that you don't outgrow your ancestry, and so the monocot-dicot distinction is within the context of being an angiosperm. If you're not an angiosperm, you cannot be a monocot or a dicot because monocot and dicot only apply to angiosperms. Then you have order, family, genus, and species. And the point of this hierarchy is to see where things fit in relation to one another. So when you have individuals of the same genus versus individuals of the same family in different genera, you know that the organisms that are in that family are united by a set of characters that everybody shares in that family. However, to be in a particular genus, you don't necessarily have to have all of the same diagnostic features as a different genus within that family. So a species should have the most key characteristics to it, whereas a domain should have the least. And so everybody in the eukarya, the only thing they have in common is that they are eukaryotic. However, when we get down to the kingdom Plantae, which is the plant kingdom, all plants have to be multicellular, as well as a few other things that characterize plants specifically. Now, it's not just the levels of domain, kingdom, phylum, class, order, family, genus, species. We have others like subphylum, subclass, superorder. We have subgenera, nothogenera, number of other things that kind of serve as placeholders or that serve as sort of less distinct categorizations, and over time these have resulted in new genera being formed or have resulted in different groups being merged together. In taxonomy, we have people who are classified as mergers and people who are classified as splitters. Some people really want to get everything in some distinctive group. Other people really want to unify everything. I am more of a splitter. The more distinctions you can find and use to characterize things differently, the better. I really like looking at differences between things. And then we'll also see things called clades. A clade is a less formal, and by less formal I mean it doesn't have to fit anywhere specific. You can have a clade within a phylum, you can have a clade within an order, within a genus even, usually not. Sometimes it's within a family, wherein you take a couple of different organisms or groups of organisms and say, they have this feature that's shared among them. They're all within this larger group. This feature doesn't necessarily put them, elevate them to the next higher level on their own. So it's a couple of different genera within a family that, they shouldn't be upgraded to their own family because they still fit within the family they're in. But there is something unique to this particular set of genera. Maybe they still are distinct enough from one another where they can't be considered the same genus. We're not going to worry too much about that, but just know that that's kind of there. So now we're going to kind of zoom in on plants. Plants are in the domain Eukarya. Like I've mentioned before, they're eukaryotic. They are in the kingdom Plantae, which includes plants. There are several characteristics that define plants, and it's important to note that algae are not plants because they lack some of these features. First of all, the organisms have to be multicellular. There are multicellular algae, but not all algae are multicellular. If you have a group where some of the individuals don't meet the classification characteristics or criteria for that particular level of classification, you have to exclude the entire group. Now, all plants are multicellular. All plants have cell type specialization, sometimes referred to as differentiation, wherein you have cells, tissues, even organs that are both morphologically and physiologically distinct. There are algae that have morphologically distinct portions to them, but they are not physiologically distinct. Or they have physiologically distinct but morphologically uniform portions, such as the gamete-producing organs, the reproductive structures in algae, which are physiologically different than normal cells, but they're not morphologically different. And finally, you need to have a sterile lining produced by cells around your reproductive structures. All plants have this, but algae do not. This basically just means that when the flowers or the spore heads are being formed on a plant, you have a group of cells that sits between the body of the plant and the flower, if you will, the reproductive portion, that prevents any bacteria, virus, fungus, any other non-plant material from getting into those cells. So the inside of a plant seed pod is sterile until the lining around it is compromised. So, you have a fruit and it's not going to rot until, something punctures the outside, or, the fruit falls and then bacteria can enter from the wound at the top of the fruit. The inside is completely sterile where the seeds are. Now, there are a couple things I didn't say here. You don't have to be green and you don't have to be photosynthetic to be considered a plant. There are a number of plants that fit these above characteristics. Now, there are a few other cellular characteristics that separate plants from animals. However, we're not going to worry too much about those because they unify the plants and the algae in a way. But you don't have to have functioning chlorophyll, and you don't have to photosynthesize in order to be a plant. We have a number of plants that are parasites on other plants or on fungus. There is actually a native orchid in Rhode Island called the coral root orchid that feeds entirely on the fungus growing from the roots of other plants, and so it doesn't produce leaves at all. It just produces its flowers and seeds. Really cool stuff. So we'll see some parasitic plants later on in this. So now we're going to get a little bit into the plant tree of life and exactly what we're looking at. So we can characterize plants as being vascular or non-vascular. The ancestral trait is to not be vascular. Plants like moss, hornwort, liverwort, and a few other things are non-vascular, where materials go between leaves through just a matrix of fluid. It just diffuses around without anything major. The advantage to this is that the plant can have fairly rapid growth. It doesn't have to invest a lot of energy into any kind of structure. The disadvantage, however, is that one, there's no way to shut off portions of the plant in the event of wounding or any kind of disease that hits it. And secondly, the plant cannot get particularly large because it can't produce enough structure in order to grow to the size of a tree. And if it were the size of a tree, it wouldn't be able to move materials around. Vascular plants, on the other hand, have these networks of tubes, veins, vasculature inside of them that are basically cell linings that allow materials to move up and down the plant. That is what a vascular plant would be. It's the difference between an open circulatory system and a closed circulatory system, if we think about animals. So humans have veins and arteries and capillaries, whereas insects, they just have a heart to slosh the blood around the inside of their bodies. Vascular plants can be broken up into seed plants and non-seed plants. Now, up until now, we have plants reproducing in a fashion where the mature plant undergoes a meiosis process where its cells get half the number of chromosomes they normally would, and then these would fall off of the plant and grow into a separate structure that would then form cells that would fuse to produce a new embryo or new, plant that's diploid again. Now, we can offset this in the seed plants. So non-seed plants would be things like ferns, mosses and hornworts and liverworts. Also, your bryophytes is what the non-vascular plants are generally referred to as, reproduce this way. Whereas seed plants, that whole process of a new haploid structure growing occurs on the plant itself, is greatly reduced, and the embryo is formed inside of a seed rather than just growing from a few fused cells that are out in the open. So seed plants do something where they fuse their cells to make a new embryo, and they enlarge that embryo to some extent in a containment device called a seed. And that seed is usually housed in something. And so these are our seed plants. Within the seed plants, we have two phyla. We have the Gymnosperms and the angiosperms. The Gymnosperm phylum is known to have a number of primitive vascular plants like cycads, ginkgo, our conifers, which are, like your pine tree and your spruce and your fir and things like that. Please note that when something is called a conifer, it doesn't mean that it's evergreen. There are not all conifers are evergreens. Not all evergreens are conifers. Not all gymnosperms are conifers. All conifers are gymnosperms, but not all gymnosperms are conifers. Conifers, gymnosperms in general, are referred to as naked seed plants because they don't completely produce a casing around the seeds. The seeds are kind of out in the open, on the plant, exposed to the air and whatnot. Whereas angiosperms make a complete covering or casing around their plant, around their seeds, normally referred to as a fruit or a seed capsule, something like that. So angiosperms are known as the flowering plants. In the Cretaceous period, angiosperms came about, and there was this huge, very important period of time known as the angiosperm radiation that occurred in the Cretaceous, where angiosperms just took off. A number of new angiosperms evolved rapidly, evolutionarily speaking, and with it, a lot of new niches or roles for other organisms opened up. And this is where we have this huge increase in the diversity of different insects and birds and a number of other organisms that are important to the angiosperms and rely on the angiosperms. And this is going to be important in some of the families that we look at, because some of the reasons the families that we look at function the way they do is because they co-evolved with some kind of animal to get something done. So when angiosperms evolved, so did a whole lot of other stuff that we know today. A lot of today's world is the way it is because of angiosperms. Most extant plants are angiosperms. We're going to look mostly at things that are angiosperms. Some of the ferns can be eaten. Fiddleheads are a thing, and people eat pine nuts and things like that, and I may do a presentation on those. But for now, we're going to focus on the angiosperms. And we have two classes, if you will, of angiosperms to worry about: dicots and monocots. Dicots are the more confusing group. They are a group that's called polyphyletic, which means numerous angiosperm ancestors gave rise to what we now classify as dicots today. It doesn't mean that there's not a common ancestor to everything in the angiosperms. What it means is that when the angiosperms first split off from the other vascular plants, there were a few groups initially, each of which on its own produced dicotyledonous plants. The initial ancestors of these groups have gone extinct since, but now we have a whole bunch of dicots that have various ancestries after that split from other vascular plants when the angiosperms radiated out. Meanwhile, they are characterized by having diffuse or branching vasculature or venation. You see, like the cross bridges between their veins and things like that. You have nodes, which are sections of the plant where material comes out of, the leaves and the stems. They can have two or more structures per node. And then we have a group known as monocots, which are a monophyletic, fairly, young group of plants. Very successful group of plants, but they are all characterized by having a single cotyledon during the germination process. Now, I should more accurately say a single cotyledon or a double cotyledon in the dicots as embryos, and that's not fully true either, but there are plants that the cotyledon doesn't actually emerge from the seed coat or from the ground when the seed germinates. Like walnuts, for example, are dicots, but you don't see the cotyledons. The cotyledons store fats and energy for that seedling, but they are not photosynthetic and they don't appear. Cotyledons are the seed leaves or the first leaves of the plant. They are present in the embryo, and they may or may not be present in the plant after it germinates. Those are those tiny little leaves on a plant when you plant your seeds that eventually fall off. the veins on a monocot are usually parallel, and they're only able to have a single leaf per node because of the way their vasculature is set up. It's almost like a half plant, wherein you don't have as many tubes and wires going through it. So that is the basics on plant taxonomy, and we're going to get into some families.